So thank you very much for inviting me. I'm going to tell you about our work on understanding genomes. Sego, Mary Jo Uyats. A little bit about me. I'm a member of the Ganawage Mohawk Band, and I just said, hello, my name is Mary Jo. This is how I looked when I was closer to your age. That's me when I was 21. These are my grandparents, Stephen Oroniatega and Bianca Skawadetsi on the Ganawage Mohawk Reservation. So how did I get here? How did I get to be a professor? Well, the teachings of my elders. I went to public schools, got a high school diploma. I was lucky to win a scholarship. I got a bachelor's degree in chemistry from Reed College in Portland, Oregon. I got my PhD at Northeastern University in Evanston, Illinois. I was a NATO postdoctoral fellow at Tel Aviv University in Israel. I've been on the faculty at Northeastern University since, but I've had visiting appointments in Denmark, at UC San Diego, at Brandeis University, at Scripps La Jolla, and at Caltech. So that's one of the nice things about being a professor. You could do sabbaticals in interesting places. Okay, so genomics. In the late 1990s, the human genome was sequenced. Oh, did I hear a question?
I think somebody was just joining in. So.
Okay, hi there. Okay, so the human genome was sequenced, hundreds of other species followed. Today, thousands of species have had their genomes sequenced, their DNA sequenced. And of course, there's this big pile of gene sequences, and what do we do with this information? Well, in the years 2000 to 2015, the United States had a structural genomics project. If we learn the structures, the argument was if we could learn the three-dimensional structures of the proteins that the coding genes code for, we would know a lot more about these genes. So over 15 years, and the US spent about $1 billion over those 15 years total, the three-dimensional structures of over 15,000 proteins have been reported and made public in the Protein Data Bank. Now, nearly all of these structural genomics proteins are of unknown or uncertain biochemical function. So the next frontier is functional genomics. How do we learn what these gene products or proteins, how do we learn what they do? Okay, so the problem is if I have the three-dimensional structure of a protein molecule, what is its biochemical function? The original plan for structural genomics says, oh, if we know the 3D structure, that will tell us what the function is. The problem is this is much more difficult than originally thought, and we've got 15,000 structures of unknown or uncertain function. Many of these proteins have functions mislabeled or misannotated. So we need an approach to function prediction in order to identify targets for drugs, and add biological knowledge to structural genomics data. Two proteins can have the same overall structure, but different biochemical function. For example, the Rossmann fold depicted on the left and the Tim barrel depicted on the right are two different possible structures for protein molecules, and each one represents over 50 different biochemical functions. So just knowing the structure doesn't tell you what it does, what is the function. So our approach is to use computed chemical properties of the individual amino acids, because proteins are made up of amino acids. There are 20 amino acid types that make up proteins. So we calculate the chemical properties of these amino acids, combined with genomic data, combined with geometric data, surface topology data, and identify those amino acids that are active in biochemical function. And then we seek a local structure match at that site, that little cluster of amino acids that are doing the biochemistry. So first we find the local site on the protein structure where the function occurs. So to do that, we invented a new machine learning method called pool, or partial order optimum likelihood, to incorporate chemical and electrostatic features that we can calculate of the amino acids, incorporate geometric features, and predict all 20 amino acid types that are involved in the biochemical function. Now, machine learning, you know about machine learning. Machine learning is where you give the computer known examples. In our case, we would say, okay, these are a bunch of proteins where we know which amino acids are functionally active. And then we ask the machine to predict which amino acids are functionally active in a protein where we don't know the answer. So you give the computer known examples and then ask the computer to predict what's true of the example where you don't know the answer. Okay, so partial order optimum likelihood, or pool, is a multidimensional isotonic regression with a monotonicity constraint, meaning that the input features to the machine are such that the outcome, in this case, probability of biochemical activity, that probability should be a monotonic function of all the input features. So we input thematics data, which are our calculated chemical and electrostatic properties, and we also can include geometric data or surface topology data. And we incorporate environment features to predict all 20 amino acid types. And this enables us to predict with very good accuracy which amino acids are involved in function. Okay, so here's an example, a protein structure called methylglyoxal synthase. The red amino acids depicted in the picture are the ones that we predict are important for the biochemical function. And in fact, that is correct. So it's not the whole protein structure. Notice there's one local spatial region where the biochemistry happens. And we can use computers to predict where that is in any given protein structure. If you give me any protein structure, I'll tell you where the site of activity is. Okay, so we can predict the functionally important amino acids in the structure. We know where in the structure the biochemistry happens. Can we use this information to predict the function? Okay, so our approach is to take proteins of known function and predict the set of functionally important amino acids. And then we perform a local alignment at the key spatial positions. And we look at what do proteins with the same biochemical function, what do their predicted local sites of activity have in common? Whatever these proteins of common biochemical function have in common at their local sites of activity, that is a signature of that particular type of activity. I'll show you some pictures in a minute. So we sort the structures into functional subclasses. We sort all of our protein structures according to their biochemical function using these local signatures. And then for proteins of unknown function, we predict what is the local site of activity in that protein of unknown function. And then we use a scoring function at the predicted key positions to assign the function. Okay, so chemicals, each functional type has a unique signature that is characterized by predicted residue types and predicted spatial positions. So for example, if the protein is a phosphoribosyl anthranilate isomerase, or PRAI, it's got a cysteine in position 1, a glycine in position 3, an arginine in position 8, a histidine in position 14, and an aspartate in position 19. And that set of amino acids in specific positions is a signature of our PRAI activity. Only indo-3 glycerol phosphate synthase, or IGPS, has a glutamate lysine combination predicted in positions 1 and 2. So they're specific types of residues at specific positions tell you what the activity is. So for example, we have a bunch of different ribulose phosphate epimerases and some structural genomics proteins that could be ribulose phosphate epimerases. And you can see we have aligned the amino acids of the knowns and the unknowns together. And notice there's a beautiful overlap of predicted residues between the knowns and the unknowns. So the green one is a representative known, and the other ones are the structural genomics proteins, and the structural genomics proteins, their predicted amino acids align beautifully with the representative known RPE. So we say, well, these are probably all RPEs because the local amino acids that are doing the biochemistry are all aligned nicely with the knowns. Okay, and then we make tables to score them. Okay, 6 hairpin glycosidase superfamily. Superfamily are a set of proteins with similar structure, but they can have different functions. So the 6 hairpin glycosidase structure has 4 known functional subgroups. And for example, these are the aligned amino acids that are important for the known glucoamylases. So look, they're all very similar structure of the local site of activity. But here is a protein from structural genomics. It comes from the bacterium Bacteroides fragilis. Bacteroides fragilis is a species of bacteria that we have a symbiotic relationship with. It lives in the digestive tract of humans, and it's one of the types of bacteria we have a symbiotic relationship with. We don't know much about them, so the structural genomics people were busy solving the structures, determining the structures of these human microbiome species proteins. And they solved this structure and they said, oh, this looks like a glucoamylase. But we took a look at it and we said no. The red amino acids are the aligned amino acids, the spatially aligned amino acids from known glucoamylases, and the blue are from that structural genomics protein. And we say there's no way this could be a glucoamylase. It just does not have the aligned amino acids, and you can't fold them together to line up with the red ones. So the blues don't, the blues are the structural genomics. They don't align with the knowns, the reds. So we say it's incorrectly labeled. Okay, so here's some other examples. On the left, there are 2 structural genomics proteins labeled as enol-CoA hydratases. And now in both pictures here, the structural genomics proteins are in magenta, and the representative known enol-CoA hydratase is in green. On the left, you see a beautiful match of residue types, amino acid types, spatially aligned between the structural genomics protein and the known enol-CoA hydratase. So we say that structural genomics protein probably is an enol-CoA hydratase. Beautiful match at the local site of activity. But on the right, there is another one. They all have cysteine aspartate lysine feature, but the other amino acids don't match at all. See how the magenta ones and the green ones don't align. They're not similar amino acid types and similar positions. So we say the structural genomics protein on the right is incorrectly annotated. Its predicted important amino acids just plain don't align with the representative known. Okay, so we make predictions, but of course we want to do some experiments to see if our predictions are right. So of course experiments take a lot longer than informatics and computation, but we have some examples. This is a protein from a domain of unknown function of a bacterium called Bifidobacterium adolescentis. That is another bacterium with which we have a symbiotic relationship. It lives in the digestive tract of humans. The structure was solved by the Joint Center for Structural Genomics out in California. And the closest structural match and the phylogenetic analysis said it was a DNA polymerase III alpha subunit. But they asked us to take a look at it, and we said no. There's a decent match with the DNA polymerase III, which its predicted amino acids are in blue, and the structural genomics proteins amino acids are in green. But note that there are two mismatches. There's a T for Y swap. It's in the dotted circle there, threonine instead of tyrosine, and there's a histidine instead of cysteine. So there are two mismatches. We said there's a far better match with histidinol phosphate phosphatase. Beautiful match. The only difference is the aspartate glutamate E189, E230 mismatch, but aspartate and glutamate both have organic acid side chains. So chemically, they're very, very similar. Those two side chains of the amino acids are very, very similar. So we said that they would do the same chemistry. And therefore, we thought that this was a hit. This was a good assignment. Okay. So we predicted it was a phosphatase, and experiments show indeed it has phosphatase activity. It was proved experimentally that it's active as a phosphatase. It was negative for DNA polymerase activity. So we correctly predicted the function. Here's another example. There were two structures from structural genomics that were labeled as ketosteroid isomerases. One was from Mycobacterium tuberculosis, which is the bacterium that causes tuberculosis, and another from Mesorhizobium loti. The Mesorhizobium loti is a bacterium that certain plants have a symbiotic relationship with. And the Mesorhizobium loti is involved with nitrogen fixation. So crops like beans, soybeans, and alfalfa that put nitrogen back in the soil, these bacteria are important for agriculture. So there were two proteins solved by structural genomics that were labeled ketosteroid isomerase. So we compared with two known ketosteroid isomerases. And we said, look, the one from tuberculosis could be an isomerase because it has the site of isomerase activity. The one from Mesorhizobium loti does not have the right amino acids at all to have isomerase activity. And so we went out on a limb and we published some time ago that the tuberculosis one was likely an isomerase, but the Mesorhizobium loti one was mislabeled. Later, and this is not yet published, we experimentally confirmed by kinetics assay that the tuberculosis one has isomerase activity, and the Mesorhizobium loti one, we were unable to detect any activity. That was measured by the conversion of the isomerization of androstenedione. Androstenedione is a steroid that athletes sometimes use illegally to enhance performance. Okay, so to summarize, we predict active residues, active amino acids in protein structures with chemical and electrostatic properties. We predict which amino acids in a protein structure are important for activity. We match these local sites and score them to predict function. And we have chemical signatures for functional types, and we look to see if the predicted amino acids in structural genomics proteins align with those predictions, those local sites for proteins of known function. We have identified at this point many structural genomics proteins that were mislabeled, have incorrect functional annotations. So chemistry and physics have important roles to play in the interpretation of genomic information because we predict the important amino acids with computed chemical and electrostatic properties. So it's all chemistry and physics. So we have some related projects. We have studied extended active sites and enzymes, amino acids far from the site of reaction. We've predicted computationally and shown experimentally that these distant amino acids that we predict to be important indeed are important. If you change them, the protein loses activity. We are also involved in the computational end of drug discovery. We have an active project seeking a way to treat familial ALS or Lou Gehrig's disease, an always fatal inherited neurodegenerative disease. We've worked on cardiac drugs, anti-cancer drugs, and now we have a very active project seeking drugs to attack the SARS-CoV-2 protein, the SARS-CoV-2 proteins from the virus that causes COVID-19. So that's a current project that we have ongoing. Okay, so these are some of the students that did the work: Ramya and Jocelyn, Caitlin and Lydia, Samantha, who is working on identifying proteins in the ocean genome that are involved in wood degradation, cellulose degradation, for the purposes of building a biofuel system. These are the people on the COVID-19 team. We use Pool to predict the targetable sites in the SARS-CoV-2 proteins and then seek small molecules that will attack and inhibit these viral proteins. So Suhasini, Kelton, Yen, and 3 undergraduates who are working with us this summer: Alyssa, Ian, and Jasmine. I thank our funders. The NSF has funded our genomics work and all of the people who've worked on this. And this is how you can reach me. Thank you very much for your kind attention, and I'm happy to answer questions.
Thank you so much, Dr. Ondrechen. That was great. Does anyone have any questions to start? If not, while you guys type in the chat, I had one question. We actually had a group in one of our projects use random forests to look at drugs that are currently on the market and then how they could possibly repurpose to like personalized genetics for cancer treatment. So looking at the cancer genome and then chemical signatures of the drug, using random forest models to predict efficacy. How did you guys do it for ALS and COVID-19?
Okay, for ALS is a different kind of approach where some people inherit a mutation in the superoxide dismutase 1 gene. And their superoxide dismutase 1 protein tends to fall apart and form aggregates in the central nervous system. Most of the neurodegenerative diseases like Alzheimer's, Parkinson's, ALS involve proteins aggregating and gunking up the central nervous system. So we are seeking a way to stabilize mutations in mutant proteins in SOD1 by basically tethering the 2 subunits together. And so that is a kind of a very different approach to drug discovery for the ALS project. For COVID-19, as you may know, the Chinese sequenced the SARS-CoV-2 gene they released it to the public in January. And February, March, the crystallographers started reporting the structures of SARS-CoV-2 proteins. So we immediately jumped on it, and we used Pool to predict the binding sites in the SARS-CoV-2 proteins. Now, the catalytic sites, you could probably guess, but there are other sites that could be important that involve other interactions where we could possibly shut down the protein or the viral life cycle. So we predicted all the potential druggable sites in the SARS-CoV-2 protein structures. And then we use conventional docking to find small molecules that could bind tightly to these sites in the SARS-CoV-2 proteins and thus stop the viral life cycle. So we joined the JEDI challenge. The JEDI was the Joint European Disruptive Initiative. And the purpose of the JEDI challenge, there were, I think, 120 or 130 research groups, teams all over the world working on it. The goal was to test in silico 1 billion compounds, billion with a B, compounds against SARS-CoV-2 targets. So we did some millions as our contribution, and we sent our predictions to the JEDI. So some of those compounds are being tested. And then my colleague Penny Boyning at Northeastern, a postdoc and a graduate student in her lab, are in the process of testing some of our predicted compounds. So the compounds will be tested first in vitro, and then promising compounds can be tested later in cell culture in animal models, and then if they pass those tests, then go on to human trials. So that's the story of our COVID-19 work.
Yeah, that was definitely really cool. We just had another question pop up. Somebody was wondering what a kinetics assay was and how that related to the steroid story you were talking about.
Oh, what's a kinetic assay? Okay, good. Okay, these enzymes catalyze reactions. For example, the main protease in SARS-CoV-2 cleaves the viral peptide chain into individual proteins. And so with kinetics analysis, you measure in the lab how fast the enzyme is able to catalyze the reaction for. So how fast does the reaction go in the presence of a certain concentration of enzyme? Okay. And then to test if, like we tested that the tuberculosis structural genomics protein was a ketosteroid isomerase, we measured how fast it converts one isomer of androstenedione to the other. So usually the product has a color that you can detect, or the product has, or the reagent has a color that disappears or something like that. So you could, by the color change, and you take time points and measure how fast does the enzyme make the reaction go. That's how you do a kinetics assay.
Perfect. And a follow-up question to that one. What's usually the time frame going from a bioinformatics discovery to actually testing it clinically, and how it actually unfolds to the public?
Okay, getting drugs to market takes a very long time. It takes years. Even for diseases where the process is sped up, is given priority, it could take years. We predict molecules in silico. They're tested in vitro. Then it usually goes to cell culture testing, and then to animal model testing. And promising compounds then go to clinical trials. Phase 1 is given to a small number of healthy volunteers to see if it's safe. And then phase 2, it's given to, very often to people who are, say, critically ill, to see if it has any effect. And then phase 3 is a larger trial with a larger number of people to see if it's effective. And then if it passes all those tests, it goes to the Food and Drug Administration for review. And that can take a while. So it's a long, long process. And, you know, for an emergency like the pandemic, some of these steps will be expedited, but, you know, nobody wants to compromise safety.
Got it. Thank you.
Professor, I have a quick question.
Please.
So in regards to the machine learning pool, does it, so from my understanding from what you said, it uses like a large database of existing amino acids and proteins in order to find similar ones. From like one use of the program, how many predictions do you usually like end up with?
Okay, pool was benchmarked on a set of 100 diverse enzymes where in the literature it's been proved which amino acids are important. Okay, and then we feed it actually 4 data points, and those 4 data points are related to computed chemical and electrostatic properties. And then pool generates then a rank ordered list where there might be 300 amino acids in the protein structure, and it will rank order them according to likelihood of involvement in the biochemistry. And then we have a cutoff criterion that we developed to decide where in the rank order list do we cut off and say, okay, the ones above the cutoff are the ones we believe to be important. So that's how pool works.
And would you say the pool technology can be used in other fields, like the fields if the code was slightly altered?
Oh, yeah. Yeah, we use it to predict important amino acids, but actually it's a general machine learning method. So it could be used for other things, as long as the outcome is a monotonic function of all the input features.
What does monotonic mean?
Like for example, prostate cancer is detected in men by PSA levels in the blood, but it's not a perfect test. There are false positives and false negatives. There are men with high PSA that they're unable to find any cancer in. And then there are sometimes men with prostate cancer that don't show these elevated PSA levels. So what you could do then is to take a bunch of different biomarkers, PSA and others, that where the level is correlated but not perfectly with prostate cancer recurrence, and then feed those data for a patient into pool and let pool predict whether, on the basis of several biomarkers instead of just one, whether the patient is likely to have prostate cancer or not. Because you don't want to put a man through the biopsy if it's not necessary, but you know, you do want to detect if somebody does have prostate cancer, and you don't want to miss if there is prostate cancer. Same thing with ovarian cancer. CA125 is the biomarker used to predict if a person has ovarian cancer. But again, there are false positives and false negatives. So yeah, pool could be used for other things. It's general, as long as the outcome is a monotonic function of the input feature.
So it's kind of like, so it's basically just giving you the probability of event occurring, basically, from what I understand.
Yeah. So it's sort of something proportional to the probability, a number that's proportional to the probability. Yeah.
Perfect. And I guess just branching off that idea, how do you guys usually determine the thresholds? For example, one of the teams this year, they were using in silico analysis of the human ITIN gene for predicting which mutations would be deleterious or not. but depending on which threshold you use, just like you said, we could be missing out upon some mutations which may in fact be deleterious, whereas others are not. So is there some established method of establishing that cutoff line?
Yeah, we wrote a paper about it a few years ago, that where we tested a bunch of different cutoffs and found a cutoff that was optimal against the data of known amino acids.
Got it. Thank you. I'll try to look for that paper as well.
I have one more question.
Please, yeah. You guys ask excellent questions, by the way.
What got you into computational bio in the first place? Did you always like math or computer science, or what led you down this path?
Well, remember, I'm 67 years old, so when I was your age, computers were not particularly powerful. And when I was a kid, there just weren't many computers at all. And certainly people didn't have their own like we all have now. But I was I liked math and I liked chemistry. And so getting into theoretical chemistry was kind of a natural path for me because I like math and chemistry. And I started off doing quantum theory, small molecules, materials, spectroscopic properties, conductive properties. And then, in the late 90s, the human genome was sequenced. I thought, gee, there's probably something that a theoretical chemist can contribute to. So I took a sabbatical. I was up for sabbatical in 2000, and I took a sabbatical in a structural biology lab to crash learn biochemistry, because the biochemistry I had learned when I was in college was no good anymore. I mean, you couldn't even sequence DNA back then. And I had to crash learn biochemistry and got interested in developing methodologies to understand proteins and genomes. That's how I got into it.
Thank you so much. That was great. Does anyone have any other questions before we wrap up?
I have one last question.
Go for it.
Where can we find any information about the progress about the JEDI project? Wait, is it called the JEDI project?
Yeah, the JEDI Challenge. Just search on Joint European Disruptive Initiative. Joint European Disruptive Initiative.
Okay, thank you.
Now they're now in phase 2. Phase 1 was for all these groups to predict compounds. Now they're in phase 2 testing of compounds. And actually there is a money prize, a significant money prize for the group with the best predictions. But we didn't do it for the money, of course. We did it because there's a worldwide emergency and we wanted to help. Oh, you guys asked very good questions. Oh, I see Adina put her hand up.
Hi, so going back to the section where you talked about protein tethering with the neurodegenerative diseases.
Uh-huh.
For those types of conditions in which I guess timing is of essence, would the tethering for the proteins have to take place at a certain stage, or would it be able to be applied at any stage?
Well, the idea is to find something that you could give to the person, because you can know through gene sequencing whether a person is going to be prone to a particular type of familial ALS. So the idea is to give the person something early, long before they show symptoms, to prevent their proteins from aggregating, to prevent the onset of disease. That's the hope. That's the idea. So what are you guys interested in? What are you kids interested in? I'm just curious, where are you all from and what are you interested in?
Go ahead. Go ahead.
Aksh and I are from California. I think we have a couple other people from our school here as well. Teresa, I don't know, you see anyone else, Aksh?
I'm pretty sure Adina and Justin as well.
And are you all interested in bioinformatics?
Yeah, at least for most of us, I think it's definitely a really pressing field because it combines, well, not only computation.
Oops, I think I lost the audio for a second there. So you guys are great.
Yeah, I think he might have frozen. Yeah, I think he's frozen.
Oh,
he's
there
back.
he's back. Yeah, he's moving again.
Oh, can you guys hear me now? I think my internet's unstable.
Yeah, we lost the audio for a second there.
Okay, yeah, so I was just saying that it's just really important to me, and I believe a lot of people, because it's applying a really growing field such as computer science and machine learning into something much more applicable and direct as biology, and you're seeing almost immediate results, something that you're not necessarily seeing if you're finding a lot of different fields, which is just what drew me into it.
Okay.
Awesome. Well, if there aren't any other questions, we'd like to thank Dr. Ondrechen for her time today, and thank all of you guys for your awesome questions. And yeah, have a great rest of your Sunday.
Okay. Thank you very much.
Thank you so much.
Bye.
Thank
Thank you.
Yeah.